Hello, my name is Thomas White.
And my name is Keizer Tumbagahan.
We are in Dr. Stephen Cowan's lab in the Department of Psychology. The lab primarily focuses on behavior and decision making.
However, at the moment, we are focusing on ketamine and its effects on the brain, specifically its effects on depression. Our question is, how does ketamine affect the brain in patients with depression? Our tool that we will be using to answer this question is the fixed array.
The fixed array works by collecting electrochemical signals from the brain. The array is implanted into a rat in a specific region of interest. The information goes from the array to the head stage, which is connected to a cable called a tether. The tether is then connected to the computer.
The data that appears on the computer are the oscillations and spikes from the rat's brain. An oscillation are multiple neurons working together, and a spike is a single neuron.
On the first day, we initiate a baseline experiment to gather the rat's brain oscillations and spikes as a control.
On the second day, we inject the rat with a specific dosage of ketamine, while the fixed array collects the oscillations and sends them to the computer. Both experiments occur over a period of 10 hours, while injections occur every 2 hours.
The data collected comes from the regions of the brain that the fixed array is attached to. The fixed array in these rats are most often attached in the ventral striatum and specifically focuses upon the nucleus accumbens.
The nucleus accumbens plays a large role in reward and reinforcement, and consequently, addiction.
But it also plays a role in motivation and pleasure, which is greatly affected in patients with depression.
We can look at the brain oscillations within the nucleus accumbens and compare the ketamine experiment with the baseline.
Through this, we can see that ketamine can affect depression. This comparison supports the idea of ketamine being used as an antidepressant.
There are many questions raised by this experiment. One question is, since this experiment was conducted in rats, how do we know that it will elicit the same effect in humans?
Also, since ketamine has the potential to be addictive, do the pros outweigh the cons in using this drug as an antidepressant?
We will continue to do more research, but for now, thanks for watching.